So welcome to my channel. So this here we will be discussing about a very important process that happens in all cells, which is so critical for the it's so important for life, right? For the living or for the organisms. For so that is DNA replication, which is a very which is the where the DNA it copies and it gives you two copies itself into into two identical copies, and in that way it transmits the your genetic information from towards the next generation, and it makes sure that whatever information that is transmitted, it is done in a very accurate manner, and there is in a very efficient and accurate manner, right? So it's very important. So this it's a DNA replication that so once the DNA gets copied and you have two identical copies of of daughter cells, then only daughter that okay then only you can then only the next cell division and all that proceeds. So and for transcription and for translation, all that process though. So DNA replication is very vital process. So let's discuss. In here we'll be discussing actually a little bit. We'll discuss the mechanism in a very simplified uh, way. We'll also discuss about some of the enzymes, and we will discuss some key properties or key factors of DNA replication. That's it. Okay, it's not a very elaborate one. It will be very little concise and short one. Okay. So let's begin. So first is what is DNA replication basically. So DNA replication is a semi-conservative. So it is a process where you basically have two identical copies of the DNA. Okay, from the original DNA double helix. Okay, so that double helix could be so it you have so one of the strands will be the parental strand. From that parental strand itself, so one of okay that will be taken. That parental strand will be taken as a template, and that will it's like a template. It gives you like a guide for the synthesis of a new complementary strand. Okay, so that's how the next strand, that's how the DNA replication occurs. Okay, that's how two copies are made, generated. So DNA replication is a semi-conservative. That means that it each of the two strands of the double helix, it serves as a template. That's what I said. It's like a template. It is important for synthesizing the new complementary strand. So it gives you all the information, all the direction, and the nucleotides which and all you have to add all that. Okay. So one of the strand will be a parental one. That's the original one, and the other is the newly synthesized one that you also call as growing strand. Okay. Okay. So this is so it will make sure the genetic information is accurately preserved and is passed on. Okay. So it occurs before cell division. It makes sure that so it is important. It makes sure that the daughter cells receives identical genome. Okay, and the process starts. Okay, so yeah. Okay, so one important. So what is so now? Why it is so important? Right, next is so it's important for cell division, as I told you. It occurs before cell division, so it's important for mitosis. It's important for the repair and all that. It's important for genetic inheritance, right? It basically this is what will be the DNA, the copies of DNA molecule. This will be then transmitted to the next, it will be passed on. It will be preserved and this will be passed on. So it is important to maintain that. It's important for genomic stability. All right. So it's actually says so the DNA polymerase. One fun fact. Okay. So DNA polymerase is a key enzyme that is responsible for the addition of nucleotides. It adds a new nucleotide. So the DNA polymerase is so fast in it and so accurate that it can actually add new nucleotides up to the rate 1000 base pairs per second, and it makes around less than one error per billion base pairs copied. So it's really fast, and really accurate as well. So we'll see how the DNA polymerase is this, becomes fast, right? What gives the speed and what makes it so accurate also, right? So let's move on. So now okay, yeah. So coming now, important aspect. Okay, so this is we'll be discussing an overview. Okay. So the double helix, which is this of the replication mechanism. So one of the things is the double helix which you have, right? That has to be first unwinded, and it has to give you the template strand. It has to expose the template strand. So that process starts. So you have to unzip the original double helix. So you basically are twisting. Okay, so you have to twist it back. Okay, like you have to twist it in a way that that they can be like they should be broken apart so that you have access to the template strand. So it is taken with analog. You take it. It's example like your ladder, right? You can imagine a twisting of a ladder tightly and then separating the two side rails, right? So those complementary bases, which it forms the rugs, right? That should be broken apart. So separation, it exposes the nitrogenous bases. That's your nucleotides which are there in each strand. So each original strand will it'll serve like a guide for synthesizing a new complementary strand. So first thing is the formation of the replication fork. So the double helix which is there, it will unwind and it will give you a Y shape, a structure, which is called as replication fork. And that replication fork, when it's created, so this is activated when the new growing strands when they are built, this fork is initiated. So since you have two original strands that are anti-parallel to each other, that means they run in opposite direction. That is, one is from 5 prime to 3 prime and the other one is from 3 prime to 5 prime. So the two template strands are in opposite orientation in your fork. Okay. So now, okay. So the daughter strand that is synthesized. So there are two things. Okay, when the synthesis happens, some things you should know. That is the leading and lagging strand concept, which is basically the strand that is daughter strand that is synthesized continuously. Okay, it's called as the leading strand. So it is synthesized it precedes the synthesis of the daughter strand. So basically, okay, it moves towards the direction of the preceding the advancing replication fork. Okay, so this is called leading strand. Okay, runs from the 5 prime to 3 prime direction. Okay, so you have to understand one thing before that the replication of DNA can only happen at from 5 prime to 3 prime direction only. Okay, so because of uh, you know this because but you have two strands, right? You have two strands that are running anti-parallel. Because of that because of that you also have like there is a asymmetric at happening that at the replication fork. So you also will have to synthesize from 3 prime to 5 prime. Okay, the nucleotides. So that's why you have this leading and lagging strand concept. So leading strand is where it's continuously synthesized. The strand is synthesized continuously, okay, from 5 prime to 3 prime. And the strand which you get to be a long smooth one. Okay, in the lagging strand the other template runs in an opposite direction. Okay, so because you can only synthesize from 5 prime to 3 prime, the okay so this okay 5 prime to 3 prime okay, see, so this runs in the opposite direction, the lagging strand. Okay, it will go in the opposite direction. So the synthesis will be it will be discontinuous. Okay, the strand synthesis will be discontinuous. It will be going away from the advancing replication fork. So as the fork opens up, like more template, okay, the machine must jump, like sort of jump, and it creates small segments. So these small segments which are synthesized, they are called as Osaki fragments, and then these will be joined together. Okay, to give you a complete single complete strand. Okay, yeah. So for the lagging strand, the direction of the nucleotide polymerization is opposite to the overall direction of the DNA chain. So the synthesis of the strand, it is discontinuous, that is it's back stitching. So it means that only that the DNA replication can happen only from 5' to 3' of the DNA polymerase. So you have short or you have transient pieces of DNA that are around 1000 to 2000 nucleotides long, which are called as Osaki fragments at their growing replication work. So uh, around okay, so 1000 and 2000 nucleotides are generated in the case of Osaki fragments. You have in the case like bacteria, prokaryotes. For eukaryotes, you have around 100 to 400 Osaki fragments that are generated. Okay, so next we'll go on to the the steps of or the mechanism, okay, of DNA replication. So there are three things. You have initiation, elongation, and termination. So it's basically you start by initiation is very basically you the okay. So DNA replication starts before cell division. We all know that. So there are certain you will first there will be specific sites which will be there in the DNA, which will which will give you which will be unwinded, and the replication will start. So those sites, right, specific sites, they are called as origin of replication. So based on the organism, there can be like few, like one or thousand. So like in the case of prokaryote, it can be like maybe one origin of replication. But in the case of eukaryotes, since it's very it's genomic, it's DNA, it's like linearly arranged, and it's more complex. So you can have more like thousands of origins of replication. You can find it in a single chromosome itself. Okay. So, for eukaryotes, the origin of replication, right? This particular this site is called as the origin recognition complex, or ORC. Okay. In the case of prokaryotes, you have something called DNA, okay, which is responsible for that. So, what happens? So, the first thing that happens first is initiation. So, you have to initiate the DNA replication, basically unwinding and basically preparing it or making it ready for the next step, that is the addition of the nucleotide. So for that, you have specialized some enzymes that is responsible for that. So you have DNA, first you have helicase, DNA helicase, which will unwind the double helix, okay, at origin, and they create single-stranded templates. Okay, so let's move on to the next. Okay, so that's what it is. Origin of replication. So replication starts at designated sites, which are known as origin of replication, or you call ORI. Okay, so uh, this basically they are recognized by initiator proteins. Okay, so in the prokaryotes, this particular it is DNA protein. It binds the complex in eukaryotes. Okay, so the origin regulation complex, which is there in eukaryotes, this will bind and mark the replication origin. Okay, these sites are the okay. So first thing is DNA unwinding. So after the initiator binding happens, the double helix is unwound, so that you can expose a single stranded templates. So here helicase. So DNA-B is the helicase for bacteria, and MCM complex is the case that is mini chromosome, something like that. Okay, chromosome complex. So MCM and DNA-B. Okay, DNA-B is for bacteria and MCM complex is for eukaryotes. These are the helicase enzymes. Okay, they are responsible for unwinding. They are recruited and they are loaded basically into DNA with help of helicase loaders. Okay, they are called as CDC. So these helicase loaders, what they do, they basically unwind they will unwind the DNA, so they break the hydrogen bond between the base pairs. Okay, between the two strands, so they form replication bubble, okay, sort of thing, and they form that replication fork, which everybody knows. So once the replication fork is formed, or you have the single-stranded DNA templates, now you have to make sure that these templates don't re-anneal and then so again become double-stranded, right? So to prevent that, you have some proteins called single-stranded binding proteins or SSBs. So they attach to these single strands. They do through something called cooperative binding. That's what they do. So they will basically this will bind to this exposed single-stranded DNA, okay, and they basically prevent the re-annealing and they protect from nucleases and all that, okay. Then next is now when you do this process, it's also important. Now when you do this supercoiling, when you remove this, when you start doing all these things, so what happens? There is the DNA tends to supercoil. Okay, so that supercoil has to be relieved. Okay, so positive supercoiling you call so to prevent that, to relieve that supercoiling, you have enzymes called topoisomerases. Okay, that is DNA gyrase in the case of bacteria and topoisomerase 1 and 2 in the case of eukaryotes. They'll also help. They'll also be there in helping in resolving this supercoiling. So the supercoiling will develop ahead of the unwinding, which is caused by the helicase. So to prevent that, the topoisomerases will also be there in resolving those supercoilings. So these enzymes all work together, okay, in that initiation part, okay, and obviously certain enzymes will also be working along the elongation until the end as well. Okay, then next part is that primer synthesis, okay. So for initiating DNA synthesis, it is important to have some kind of what do you say, a start. So to start the initiation, you need a short stretch of nucleotide. Okay, this are so the, to which is used like for initiation, right? That is primer. So you use RNA primer, short RNA primers are used, okay, which will be complementary to the DNA template. Okay, so around 10 nucleotides this will be this primer, what is it? So that how this primer is synthesized, RNA primer, specifically it's RNA, right? So how do you synthesize RNA primers, small, short segment? So it is done by a enzyme called primase. So primase will basically synthesize a short RNA primer, which is around 10 nucleotides approximately at the start or at the origin, and they give you free a 3' hydroxyl. So only at a free 3' hydroxyl can the initiation happen. Okay, the DNA, your DNA nucleotides can be added only at the at a free end of the 3' hydroxyl. So you should have a 3' free 3' hydroxyl group so that you can add the nucleotides, right? So that is this. So that initiation that is provided. So primase basically will synthesize this short RNA primers, which are complementary to DNA template. They'll give you this free 3' hydroxyl, okay, which is needed by the DNA polymerase to start the synthesis. So the initiation makes sure that DNA polymerase is prepared. It's ready for the addition of the nucleotide, and it can do it and there's nothing there that it will stop it from doing it. So it can just go on with the addition of the nucleotide. Okay. So that is the whole initiation process. It's just to initiate, it's to give you a starting point. It is to give you that specific site where you have to initiate, and the enzymes, that is helicase, which helps in the unwinding. Okay, helicase will be there function till the end because it has to keep unwinding, right? It also helps, and then you also have single-stranded binding proteins, which are help in preventing the reannihilation or secondary structure formation. And then you have topoisomerases, which are DNA gyrases, which will help in resolving supercoiling that forms ahead of the helicase activity of unwinding of the DNA. And then you have primase, which is the enzyme that synthesizes short RNA primers, which will give you like a start. Okay, it provides the 3' OH, 3' OH end. So this will give you it gives like a start for the DNA polymerase to start adding the nucleotides. Now let's move on to the next, that is elongation. Okay, so the DNA polymerases add the new DNA nucleotides to the 3' end of the growing strand. Okay, so synthesizing DNA in the 5' to 3' direction. It's based on the complementary strand, complementary template strand. Okay, so the leading strand is synthesized continuously towards the replication fork. It requiring just a single RNA primer, right? So the lagging strand is synthesized discontinuously. Okay, it is away from the fork, right? So it creates, so you have short segments called Osaki fragments. Each Osaki fragment begins with an RNA primer synthesized by primase. Okay, so the DNA polymerase then extends these primers, and the DNA ligase enzyme will come and join these Osaki fragments. So the unwinding of the DNA helix, okay, okay, so the DNA synthesis happens, leading strand, lagging strand, you have Osaki fragments. Once the Osaki fragments are all they are sealed, the primer will be removed. Okay. So the DNA polymerase extends to Osaki fragments. The primers are removed, RNA primers. They are removed through exonucleases. Okay. So and then the resulting gaps are then filled with your DNA polymerase, and then the seal, any kind of seals or nicks that are there, or any gaps that are there that are there in the phosphodiester bond, those are sealed by the DNA ligase. So this will give you a continuous strand. Okay. It also makes sure the processivity of the DNA polymerases, which is basically uh, holding the polymerase into the template for efficient elongation. So this is done by sliding clamp proteins, which are present. So this is the first thing. So first is primer synthesis. Okay. So first step. So primer synthesis, what happens? Basically, you have your RNA, your primase, which synthesizes short RNA primers, which are complementary to the single-stranded DNA. It gives you a free 3' OH for the DNA polymerase to start. Okay. Then you have DNA synthesis by DNA polymerase. So the DNA strand synthesis happens. So the DNA polymerase will start adding nucleotide to the template strand in the 5' to 3' direction. Then you have the leading strand, which is, so the leading strand, the DNA polymerase will start here. It will synthesize continuously. It will be synthesized towards the replication fork. Okay. Then you have the lagging strand, which will be it will be synthesized discontinuously. It forms Osaki fragments. Each fragment will start with a new RNA primer. It will be in the opposite direction, basically. Okay. The next is primer removal and gap filling. So after the DNA polymerase have extended the Osaki fragment, the RNA primers will be removed. Okay, by enzymes like exonuclease. Okay. We'll discuss in the termination part. So those gaps will be filled by the polymerase. Then the nicks or gaps which are there between in the phosphodiester bond between the template, between the nearby Osaki fragments, they will be sealed together by DNA ligase, and they will create a continuous strand. Then the processivity also is maintained, that is sliding class processivity, in the sense the how many the rate at which it can the DNA polymerase can add the nucleotides to the growing strand. Okay, the speed basically. So that speed is maintained by certain proteins like sliding clamp proteins, which are example like PCNA in the case of eukaryotes. They'll help in holding the DNA polymerase onto the template for efficient elongation. Okay. So this is a whole little bit figure here. You can see this is the new strand, the orange color. Okay, and the okay, you can see the orange one. This is the leading strand. Okay, and this is the lagging strand, the blue one. Okay, And you can see the DNA polymerizes. Okay, this is the helicase. So helicase helps in the unwinding. Okay, so this is, so you see the in the lagging strand. Okay, and leading strand. Leading strand does it is synthesized continuously. In lagging strand, it will be, you will have, it will be like, the RNA primers in between the primers have to be added. So you basically get short stretches, short fragments of DNA you'll get, that are your Osaki fragments. Those fragments will be sealed together by your DNA ligase, okay, and they will be filled by your gaps and all, which will be there, that will be filled by DNA polymerase. Okay, then you will get a continuous strand. Leading strand is synthesized from 5' to 3', and the lagging strand is from 3' to 5', so opposite direction, right? That is why there is a little difference in the synthesis. Now, two things that you should know that are important when it comes to DNA polymerases. One is processivity. What is processivity? Basically, uh, that is facilitated. Okay, that is the sliding. Uh, uh, okay, sliding ability of the DNA polymerases along the DNA template once it's bound to a primer template junction. So DNA polymerases, they interact tightly with the double stranded portion of the DNA, okay, in a very sequence non-specific manner. So they are, okay, so what are this basically processivity? Processivity is basically, it is the average number of nucleotides that you can add each time the enzyme will bind to the primer template junction. So it will bind in a non-sequence non-specific manner, okay. That means it doesn't require a specific sequence for it to bind, okay. So each DNA polymerase has its own characteristic processivity that make sure it's how fast it can elongate or synthesize. Okay. So the rate of synthesis the is, it is increased when the nucleotide adding of the multiple nucleotides per binding unit, if that is increased, okay, or if that is fast, then the rate of synthesis of the DNA is also pretty much fast. Okay, so that is one of the why I have said before that the 1,000 nucleotides per second base pairs, right? That shows how fast the DNA polymerases are in addition of the nucleotides. Okay, so that is actually pretty good things. That means you can add more number of bases at a very short given of time, right? So it doesn't take a long time, which is pretty good. And then you have another factor, which is, so what if your DNA polymerases, they make very less mistakes, but if they can make mistakes, so they can make approximately one mistake in every 10 to power 10 base pairs added. So if there is a mismatch is there, then the DNA polymerase will not proceed forward. Okay, it will not add, if there is any mismatch is there, then it will not add, it will decrease the ability of adding the new nucleotide. So what happens, the DNA polymerase has another site which is there, other than the you know active site. You have a site which is for the exonuclease, basically, okay, where it basically removes the incorrect base pairs if they are added. Then that will be removed by a kind of nucleases called as proofreading exonuclease, which are enzymes basically that degrade the DNA starting from 3' end, 3' DNA end, okay, that is from the growing end of the new strand. Okay, so the mismatched nucleotide is there, that is removed. Okay, it is done by the reduced ability of the DNA polymerase to add the nucleotide adjacent to the incorrect base pair. Okay, so the this okay, so this the so basically, if the when the nucleotide mismatched nucleotide is added, it decreases the rate of the nucleotides which are added. Okay, that is, it decreases the active site which is responsible for the addition of the nucleotides, and it increases the proofreading activity or proofreading site, exonuclear site of the enzyme. That the same DNA polymerase is the one that does both the addition and the exonuclease activity. So that is one of the specialty of DNA polymerases. It can do both 5' 3' addition of nucleotide and it can do the 3' to 5'. It can do the exonuclease activity also. So that is okay. It can do 3' to 5', 5'. Okay, that activity it can do. Can add. It can do exonuclease activity as well. Okay. So the same enzyme that is responsible for the addition itself will gets this has the second chance to repair it or remove that incorrect base pair with the help of the exonuclease. That's why this this activity is present. So if there are any incorrect base pairs are there, okay, then when the nucleotide returns to the correct state, the incorporated nucleotide is mismatched with the template. It should be removed. So for DNA synthesis, so proofreading occurs without releasing the DNA from the polymerase. So if a mismatched base pair is there in the polymerase active site, the primer template junction is destabilized. It creates several base pairs of unpaired DNA. Okay, so the DNA polymerase will active site. It binds in such a way that the mismatched template okay, it is poor. So it is removed by exonuclease activity. Okay, so the newly unpaired 3' end, it moves from the polymerase active site, it goes to the exonuclease active site. So the incorrect nucleotide is removed through the exonuclease activity. Some additional nucleotides also will be removed. The removal of this mismatched base allows the primer template junction to reform and rebind the polymerase active site and allowing the DNA synthesis to continue. Okay. Next is the termination. So the termination phase of DNA replication completes the replication process. So it resolves the replication forks, filling the gaps, and resembles. It helps in deassembling the machinery. Okay, separating the daughter DNA molecules. So the following detailed steps are there for termination. So first is replication of the fork convergence. So the convergence replication forks, which so they'll meet oppositely, they meet and they complete the new DNA synthesis at the termination zone. So often they are marked by certain specific terminator sequences and proteins like TUS, which is there in E. coli for prokaryotes. The next is removing of the primer and gap filling. So RNA primers have to be removed. They are removed by exonuclease activity. Okay, that is done by DNA polymerase 1 in the case of prokaryotes and RNase H, which is a nucleus basically in eukaryotes. Okay. Then you have the next, then in the case of gap filling, so DNA polymerase 1 and pol uh, delta or epsilon, they are polymerized. They will help in the filling of the gaps. Okay. Uh, then the next sealing is there. So DNA like ligaments. Okay. The DNA ligase, which is there, that will help in sealing the gap and that help form the phosphodiester bond. Okay. The next important thing is, so the first thing that concerns resolution is of topological stress. So when the unwinding of parental duplex happens, it leads to the overwinding of the unreplicated DNA. So it leads to the formation of positive supercoils, which are there ahead of the fork. So if there are too many supercoils accumulate, then further unwinding becomes energetically unfavorable and the replication can stop. So the way this is removed, so you have to remove this or you have to relax this supercoil, positive supercoil, and that is done by topoisomerase type 1 and 2. Okay, they relieve the supercoil. Okay, they basically introduce transition break. Okay, single or double stranded break they'll introduce in the DNA strands. Okay, so it basically introduces snake-2 supercoil and helps in the unwinding and compaction of the DNA during replication. Okay. The DNA and also the topoisomerase 4 and 2, they help in the decatenation, which is basically the process of untangling and intertwined DNA, which is there after replication. So this is important for circular DNA for bacteria and all. Okay. Then the last one is replisome disassembly, which is basically removing all the enzymes, all replication machinery that are involved in this replication process, there all those proteins enzymes are removed like helicases. So the combination of proteins which basically function at the replication fork is referred to as replisome. Okay, so this is the whole termination step. And now these are the enzymes, important enzymes. So first I told you DNA and OREC. These are the enzymes or sites that helps in the recognition of the or recognition of this origin of replication basically. So DNA is there in prokaryote. This is ORC is in eukaryote. Okay, its function is basically to bind to the origin of replication and initiate unwinding basically. Then you have helicase. Helicase is DNA B for prokaryotes and MCM complex for eukaryotes. Helicase helps in the unwinding of DNA double helix at replication fork. They have the single-stranded binding protein, which are there in both prokaryote and eukaryotes. They help in stabilizing and protecting single-stranded DNA. Then you have DNA gyrase and topoisomerase, topoisomerase 2, 1 and 2. Okay, so DNA gyrase is for prokaryotes and topoisomerase 1 and 2 is for eukaryotes. So they help in relieving the supercoil and resolving tangling. Then you have primase, which helps in synthesizing short RNA primers to initiate DNA synthesis. This is there in both prokaryotes and eukaryotes. Now coming to polymerases. Now DNA polymerase 3 and 1 are present in the prokaryotes. These are the enzymes. So polymerase 3 is basically important for the main part, that is synthesizing in the making the basically the elongation, synthesizing new DNA strands. DNA polymerase 1 is primarily responsible for removing the RNA primers and replacing them. So this is important. So DNA polymerase 3, how the difference, though the function is same, but the DNA polymerases, the types are a little different in prokaryote and eukaryote. So that you should know. So DNA polymerase epsilon and delta. This is the case. This is for eukaryotes. And 3 and 1, they are for prokaryotes. So DNA polymerase epsilon and delta, again they do the same function. So the DNA polymerase epsilon functions in with synthesizing the new DNA strand in the leading strand. The epsilon is responsible for synthesizing the lagging strand because you have two strands there, right? That's why. So two polymerases are involved here, basically. Then other enzymes are RNA-SH. Okay, RNA-SH is, as I told you, they're involved in the removing the RNA primers. Okay, they're there in eukaryotes primarily. Prokaryotes, as I told you, it is the DNA polymerase 1 that does this function. Then you have DNA ligase. They're in both. They help in sealing the Osaka fragment and help in phosphodiester bond. Then you have sliding clamp, which is proteins which are present. It helps in increasing the processivity in DNA polymerases. Then you have DNA isomerase 4, which is there in prokaryotes. This helps in removing the decatenating circular DNA. Okay. Now another concept of when it comes to so that okay. So okay. So and so this is the whole concept of DNA polymerases. Okay, DNA replication and the enzymes involved. So this is the diagram it shows here. This is for eukaryotes, basically. So this is the pol A. Okay, two. Okay, basically any two. Okay, one is for C. This is for the uh, lagging strand. Okay, or Osaka fragments. Yeah, this is a leading strand. Okay. Also, you're seeing the topoisomerase here. This help in the relieving supercoil. Okay, you have the DNA primer, the primase, which will add the primer here. Okay, and then you have basically helicase. Helicase helps in unwinding. Okay, of the DNA double helix, exposing the single strand. Then you have single strand binding proteins. This is just a diagrammatic representation. So I hope everything is clear. This is the whole part now of replication of DNA. It's so important. This is just a very simplified version. It's not very elaborate. Okay, so So this is to give you an understanding about what it is in a very simple way and all the enzymes that are involved. Okay, it's pretty much complicated process in the real this, but this is a simplified version way of telling. Okay, so these are some books which you can refer. That is Leninger, which is pretty good actually. It gives you a little bit complex of complexity in terms of the biochemistry as well. It Molecular biology of gene. This is good. This is a book that I have referred also. This is pretty good. To know more complex part, to know more about the enzymes which are involved. This is pretty good. Okay, and genetics. So these are some books which you can refer. Okay, so I hope you guys all love this, like this channel, like this video basically. So yeah, more will be coming from this channel. So keep in touch.